All right, thank you. Um, my name is Giles Ross, and today I'll just be talking about my work at the Netherlands Institute for Ecology. And the title of my talk is Integrating Soil Biodiversity into Ecosystem Services and AI Approaches. And I'm working on the SOB 4ES project, which is the Horizon 2020 project, funded by the EU and the Swiss and UK governments. So just about the concept of soil health, we've all seen this Venn diagram whereby physical, chemical, and biodiversity aspects are really important for the contributions of multifunctionality and the ongoing sustainability of ecosystems. But it's often that biodiversity is overlooked because of difficulties in identification, cryptic speciation, and few morphological differences. So it's a really important aspect that we can apply novel technologies, including sequencing and machine learning algorithms to help us really get an idea of the diversity and community structure of soil biodiversity. And so soil biodiversity, from the largest macrofauna, including earthworms and ants, to mesofauna, such as mites and springtails, and the microfauna, the semi-aquatic nematodes and anchitrades, all work together with microbes, bacteria, and fungi to perform the functions that contribute to resistance to disease, resilience to environmental change, and redundancy in case of biodiversity loss. And so just to clarify, the services that they provide, that we benefit as humans, are all part of their processes that they contribute to, such as soil structure that facilitates macro and micro porosity, that influences water holding capacity, and functioning for reducing floods and other effects, hydrological, and also including carbon storage and carbon cycling, whereas up to 3 times the amount of carbon is actually stored in soils compared to all plants and animals and in the oceans above the ground. And finally, but most importantly, the decomposition and nutrient turnover that they contribute to, essential for above ground plant growth. So capturing the contribution of soil biodiversity is really an important part of understanding soil health. And in doing so, we'll also contribute to better indicators and benchmarks across land use types that are involved with the European Union Soil Observatory biodiversity dashboards, where they've only got one indicator at the moment, I think based on eDNA, but we'll also be able to verify these environmental DNA assessments that are being carried out by Lucas 22 and AI for Soil Health projects. And additionally, the EU Soil Monitoring Directive and the Food and Agriculture Organization biodiversity monitoring requirements will also benefit from more robust indicators and benchmarks of soil health using biodiversity, which is what we're trying to do in our project. So the project's subforce soil biodiversity into ecosystem services, or subforce, I like to call it, soil biodiversity, a force to be reckoned with. And we're really hammering this problem by using standardized protocols to make sure comparable data across sites and land use types, and also taking into account spatial and temporal variation by having a set of samples stretching from across the EU, 9 pedoclimatic zones from northern Scandinavia down to southern Sicily, and from Ireland in the west to Romania in the east. And those land use types, forest, grassland, arable, and lesser sampled urban and wetland sites will be also sampled across an intensity gradient of low, medium, and high management practices, whilst a subset will have been resampled an additional 3 more times in the map on the right, where you can see the subset of sample sites. And so to really get to grips with how to monitor and implement the monitoring components required for the directives, we include soil descriptions and detailed soil type and management information that will contribute to rich metadata, alongside the physical and chemical parameters and a comprehensive soil biodiversity assessment. So the 3 main components. But for the biodiversity assessment, first we extract the animals from the soil, and using a combination of morphological and molecular identification approaches, we hope to apply these and use machine learning to assist with abundance and density measurements based on image capture, and also using sequencing and high throughput sequencing to really unpack the community structure and diversity matrices. And the identification using sequencing, also combining information directly sequenced, extracted from animals, as well as the environmental DNA samples, to really get to grips and have a level of calibration to understand the sequencing outputs in terms of what's actually in the ground. And so, as I mentioned before, machine learning can contribute to abundance and biomass, which is important for linking below and above ground vegetation and parameters, and reducing the time taken for image analysis, perhaps from half an hour to count one sample to just a few minutes. And furthermore, diversity and community structure with food web networks, with machine learning unpacking the complex networks that go into structuring below ground communities. And finally, unraveling the links with environmental variables and abiotic parameters to really hone in and focus to identify those biotic indicators and targets that might be involved with contribution to ecosystem services and those links to soil health. And so our next steps would be to really promote the widespread inclusion of biodiversity sampling and more comprehensive sampling using standardized protocols to get a really clearer idea and build up our repositories of information for soil biodiversity, for morphology and molecular databases, and also for images for further training of those machine learning algorithms. And these will contribute to finding appropriate and sensitive indicators for inclusion in the routine monitoring that we all hope to contribute to for soil health. And just acknowledging the Subforce Consortium with the coordinator and Wim van der Poot and Justine Lejolie at NIOO, and some other Subforce partners there, and all the participating institutions in the consortium. And thank you very much.